Welcome back. This is part 4 of our video series on cell signaling. Last time we talked about receptor activation in terms of it being the first stage in in cell signaling. We talked about receptor activation, signal transduction, and then cell responses. So we're going to go back to the beginning and we're going to look at reception of the signal. We're going to look at 4 types of receptors in this video, 3 of them on the cell surface, and then 1 we'll look at inside the cell, so-called intracellular or cytoplasmic receptors. And again, don't forget, this is general, so there are thousands and thousands of specific examples of these receptors, and we're just talking in general terms about the mechanistic processes by which information is received by cells, and later we'll talk about signal transduction processes. Okay, so the first type of receptor we're going to look at is called an enzyme-linked cell surface receptor. So this is found on the cell membrane, and so we've got our cell membrane here, and then we've got the receptor, and this is very similar to what we saw last time in our last video, and here is our signaling molecule. So our signaling molecule there is bound to the receptor, and the receptor is being turned on. So these receptors have transmembrane regions, which are the regions which span the membrane. They have the signal binding or ligand binding domain, which is on the outside of the cell, and then inside the cell, the enzyme-linked receptors have a domain which is an enzyme, and that enzyme is often called a kinase. And so let's look at what a kinase does. When a kinase becomes activated, it will take the phosphate group from ATP and transfer that onto another protein, which turns that protein on or off, and that's the start of signal transduction. So let's see what's going on here with this enzyme-linked receptor. Here's our inactive receptor, and there's no ligand present, so there's no information being transmitted to the cell. Inside the cytoplasm is a protein which is going to have its behavior changed in response to receiving the information. So this is the first protein in the signal transduction process here. So no receptor, and then this protein is said to be unphosphorylated. And what we're going to do in a moment is add a phosphate to it, and that will make it phosphorylated and turn it on. So along has come our signaling molecule, our ligand. It's bound to the extracellular ligand binding domain. That information has been transduced across the membrane to this kinase domain of the receptor. So that's a set of amino acids folded up, and the behavior or the function of that set of amino acids in that domain of that protein is to take a phosphate from ATP, and it's going to be this terminal phosphate here, this last phosphate here. The kinase will take that phosphate and will put it onto this protein here, and that was our unphosphorylated protein, and now that's become phosphorylated. This is what kinases do. They take a phosphate from ATP and stick it on another protein or a lipid, and that changes the behavior of that protein or lipid, and here it's a protein, and so that protein will now go and do something in a signal transduction cascade. It'll send the information on in the cell. So that's called an enzyme-linked receptor. Our second type of receptor is called a G-protein coupled receptor. So let's take a look at what happens with these. Once again, we're at the cell surface. So we're going to have a receptor. Here it's called the G-protein coupled receptor, and for short that's abbreviated to GPCR, so we don't have to keep saying that long name, G-protein coupled receptor. So there's the receptor. Up here is our ligand, and we'll look at what happens with that in a moment. And down here is the G-protein, which gives the G-protein coupled receptor its name. So the G-protein is going to work with the receptor. G-proteins have 3 parts to them, so they are heterotrimers, and so those are 3 proteins that have come together, and so that's another nice example of quaternary structure. So we have an alpha subunit, a beta subunit, and a gamma subunit, and so those are 3 separate proteins, and they all work together in the G protein. So what happens in this state is nothing. When the ligand is not bound to the receptor, so here's the ligand and it's not bound to the extracellular ligand binding domain, everything is switched off. The G protein isn't doing anything. The alpha subunit of the G protein binds to GDP when the receptor is turned off. So the receptor is off, the alpha subunit of the G protein has GDP bound. Now GDP is like ADP. You're familiar with adenosine triphosphate. That's adenosine with 3 phosphates on it. You can also have guanosine diphosphate. That is guanosine with 2 phosphates on it. You can also have GTP. We'll look at that in a moment. Okay, so let's bind the signal, or the ligand, to the receptor. Now, just like we saw with the enzyme-linked receptors, that causes a structural change to be transmitted across the membrane in the protein, and this part of the receptor changes shape then, and that causes the G protein to get attracted to the receptor. So now the G protein is bound to the receptor, so they're stuck together. It's not irreversible, they're loosely stuck together. Now, as soon as that happens, the alpha subunit, which is bound to GDP, lets the GDP go. So here is the GDP being let go. And the alpha subunit binds GTP. So here is GTP being bound. So that's guanosine triphosphate, guanosine with 3 phosphates. So this is like a little molecular switch. No ligand bound, receptor is off, G protein has GDP bound, receptor gets activated by a ligand, GDP is released from the G protein, and GTP gets bound. And what happens then is that this alpha subunit separates from the beta gamma. So here is the beta gamma subunit stuck together over here. Here is our alpha subunit. And alpha can go off and do something, and beta and gamma can go off and do something. So again, that's usually the start of a signal transduction response. So how do we switch this off? It's really important that you switch these pathways off, because if you switch them on and you just leave them on, that could lead to things like uncontrolled cell division, and that's the kind of thing that goes wrong when you get cancer. So it's important to be able to regulate these things by switching them on and switching them off. So fortunately, what can happen is the ligand will let go, and that just happens because the ligand binds and lets go and binds and lets go. Proteins don't stick together irreversibly. They can stick together and then come apart, and there's a lot of biochemistry behind that we're not going to talk about. But that doesn't change the alpha subunit of the G protein is still bound to GTP. As long as the G protein has GTP bound, it is sending information into the cell. So we can let the ligand go from the receptor, but as long as the G protein has GTP bound, we're still sending the information into the cytoplasm, and that can be a bad thing. So how do we switch this off? Well, this G protein alpha subunit has got a built-in timer. So after a certain amount of time, the GTP will be hydrolyzed to GDP. So we take one of the phosphates off of GTP, and that leaves us with GDP. And so that switches the alpha subunit back off again, and the beta gamma come back together with alpha, and we reset and go back to where we started from, which is over here. So that's how we switch this on and off and on and off. Okay, so there's a couple of types of receptors. Let's look at some of the questions in the kind of style that I'm going to ask you when we take quizzes and things like that. So here is a question. I'll leave it with you, and I'll pop the answer on the screen shortly. Okay, here comes the next question. And here's the last question in this series of 3. Okay, let's go on to our third type of cell surface receptor. This is relatively straightforward. There's a type of receptor called a ligand-gated ion channel. You can find these on the surfaces of cells. All cells have these. You can also find these inside cells on internal membranes as well, but we're going to just focus on those which you find on the cell surface. So like before, here is our membrane, And to get this channel open, it's also got a receptor associated with it as well. So part of the protein here is a receptor, and so these parts here on the channel can bind to specific signaling molecules. These signaling molecules are really specific. This one in green will only open this channel. It won't open any other channels. It won't activate any G-protein coupled receptors. It won't activate any enzyme linked receptors. These signaling molecules are specific. So if a cell doesn't have the receptor, it can't respond to a signal. So this is one of the ways that you get specificity of response. Some cells need to divide, others don't. Those that do have receptors that will help them make the appropriate choice. Other cells, they just don't express those receptors. So ligand binds to the channel, and this channel has got 2 ligand binding sites, and when that happens, the channel will just open. So this is like a lock, unlocking a door. When the door's unlocked, it can open. So the channel springs open, and then these channels let ions flow down a gradient, and so as soon as the channel opens, these ions come flooding into or out of the cell, depending on which way the gradient is. In this case, they're coming down, which looks like to be into the cell. So that's a ligand-gated ion channel. This is really important in biology because it lets cells be excitable and respond by generating changes in membrane potential, which is important in generating nerve impulses. So that's our third type of receptor. The last one we'll look at here before we wrap up is called an intracellular receptor. So we're going to use a concrete example here. The estrogen receptor is probably one of the best studied intracellular receptors. So these receptors are inside the cell, sometimes in the cytoplasm, sometimes in the nucleus. And so the ligands for these receptors have to be able to move freely across the membrane. Otherwise, they can't get the signal from outside the cell to inside the cell. So let's look at what happens with this one. So the ligand is going to be estrogen, and estrogen is being produced in the ovaries. It's being produced by adipose tissue. And so the estrogen can move freely across the membrane. And so it can do that, remember, because it's sufficiently small and it's hydrophobic. So as long as there's a gradient, estrogens have a net direction of movement into the cell. And so it can also then move into the nucleus through a nuclear pore, for example, but it could also diffuse across the nuclear envelope, I guess, as well. Once it gets into the cytoplasm there is a protein, the estrogen receptor, that is held in an inactive form. And the estrogen receptor only becomes active when estrogen binds to it. So estrogen binds to it, and it changes shape upon estrogen binding. 2 of those receptors will then come together, and the function of those dimerized estrogen receptors is to change the expression patterns of certain genes. So some genes will get switched on, and that will be important in producing mRNAs and proteins which change structure and function in the cell. So this could be important during differentiation, it can be important during cell growth, and during cell division. So that's a quick example of an intracellular receptor. So that gives us 3 types of cell surface receptor: the enzyme linked receptor, the G-protein coupled receptor, and the ligand gated ion channels. And then our 4th one here is an intracellular receptor. So don't forget, we're making generalizations, except for that last concrete example. These are general ways in which these things work. When you're studying this, make sure you understand what would happen if we interfered with the function. So if I broke one of these receptors, could you predict what would be a general effect of that mutation, which would break the pathway in most cases? So I'm going to wrap up there, and I will see you next time for our last video on cell signaling.